Hello guys, welcome to my YouTube channel. I am Mampi, your favorite life sciences teacher. Thank you for tuning in. Please subscribe, like, share and comment. Do not forget to turn on the notification bell so that you can be notified every time I post a video. I hope you guys are really enjoying this lesson, and today's topic is going to be DNA replication. So far, if we check our prior knowledge, we've learned about DNA, we've learned about nucleic acids, nitrogenous bases, the shape of the DNA, and also the differences between DNA and RNA. So today we are going to learn about DNA replication. By the end of the lesson, I want you to know what DNA replication is, why is it important, and where and when does it take place. Those are the things that we need to ask ourselves during every lesson or at the beginning of every lesson. Now, before I continue with DNA replication, I want us to look at the helical structure of DNA, because previously we just saw how it is double-stranded, but we never saw the helical structure. So basically, when we say it's helical and it's winded, it's more like it's like this. So that is a winding. It's a very long structure that is coiled and winded, and we call it double helix. And then we say it's double helix because it has both strands from the left and also from the right. If you can see from my structure, adenine pairs with thymine through 2 hydrogen bond, and then guanine pairs with cytosine through 3 hydrogen bond, and then T represents thymine, A represents adenine, guanine is represented by G, and cytosine is always represented by C. These are the things that I need you to understand before we can do the whole process of DNA replication, because it's going to make you understand how the whole process works. To answer the first question is, what is DNA replication? DNA replication is the process whereby one DNA molecule makes the exact copy or the identical copy of itself. So we start with one DNA molecule, but we are going to end up with 2 identical copies of DNA molecules. So if they ask you, it's usually 2 marks. If they ask you what is DNA replication, then you know DNA replication is the process whereby the DNA makes the exact copy or the identical copy of itself. So basically, whatever that is happening, everything, if we start with AGTC, when we are going to end up with DNA replication, we should have the same structure. So we are going to have 2 of this DNA molecules. So that is basically the DNA replication process. Another question can be, where does DNA replication occur? Or they might ask you, where does this process occur? And then you're going to say it occurs in the nucleus. Remember, we spoke about the location for DNA. We have nuclear DNA, mitochondrial DNA, chloroplastic DNA. However, now we're going to talk about the nucleus. So the DNA replication takes place in the nucleus. And then another question, another important question is when does it happen? So it happens during interphase. Of course, now you may not know what interphase is, but we are going to look at it in the 3rd or the 4th lesson when we are discussing the cell division processes, which is meiosis and also mitosis. So DNA replication occurs during interphase, and then it takes place in the nucleus. And the aim, the reason why we should have DNA replication is to double the genetic material. Remember from the previous lesson we said DNA carries genetic code. So in order for us to have more genetic material, the DNA needs to replicate itself so that we can double that genetic information. So now you know what it is, where it takes place, when it takes place during interphase, and then you also know its significance. Its significance is simple: to double genetic material. Now, they'll usually ask you to write down the process of DNA replication. They might tell you that the DNA replication is the process where DNA makes the exact copy of itself, or they might ask you, what is the process that the DNA makes a copy of itself? And then you say DNA replication. The second question will be, write down the process mentioned above. which is DNA replication. I've included the structure that illustrates DNA replication. I've also included the notes, so it's up to you on which one, but I want you to understand both. If you understand this one, obviously you're going to understand this one also. So the first thing is, since when we said DNA is a double helix and it's coiled, so the first thing it needs to do is to unwind. After unwinding, the weak hydrogen bonds need to break, and when they break, the strands will do what? Will unzip. After unzipping, we are going to result with two separate strands. After unzipping, we are going to have two separate strands. The separated strands are going to act as a template to form new strands. So what happens is, a template is a guide for the formation of new strands. So we have this strand, we also have this strand. Now, when we want to form the new strand, these ones are going to act as a guide. And what do we mean when we say a guide? We know that adenine always pairs up with thymine. Guanine always pairs up with cytosine. So what you're going to get is that from here, this is going to act as a guide to say that, okay, since when we have an adenine, we need thymine, we need cytosine, and we also need cytosine. On the other strand, since this one pairs up with adenine, we're going to have adenine, we're going to have guanine, we are also going to have guanine. So what will then mean is that we are going to have new strand or new double strand formation. So after separation, it acts as a template for the formation of the new strands. After that, we are going to have the free nucleotides. The free nucleotides are the ones that are going to form the new strands using the complementary bases. And then we are going to end up with two identical copies of DNA. We started with one. Now we are going to end up with two. That is DNA replication. If you look at the process, we say double helix unwinds. I need you to know this by heart. The first step of DNA replication, double helix unwinds, weak hydrogen bond breaks. When weak hydrogen bonds breaks, they cause the strands to unzip. When the strands they unzip, they result in two separate strands, which will act as a template. Now your template is a guide for the new strands. So when we have our templates, the free nucleotides form new strands. We are going to end up with two identical DNA molecules. I hope it makes sense. And then we also have one important enzyme, and that is DNA polymerase. DNA polymerase controls the whole process of DNA replication. If they ask you what is the enzyme that facilitates or that controls the whole process of DNA replication, then you know it's DNA polymerase. That is the enzyme that controls this process. I want you to imagine me as your favorite teacher. You imagine me as a DNA. So basically what happens is, this is the DNA, and then when it's coiled, it needs to uncoil. Now I've uncoiled, it's me individually. What is going to happen is the cells which are holding me together, or the organs, are going to split into two. When they split, we say weak hydrogen bonds, they break and then they unzip. So if I unzip, the other half will go to the other side, the other half will go to the other side. In order for me to be complete, on my left side, I need the right side to complete me. On the right, I need the left side to complete me. And then you end up with two life sciences teachers. Wouldn't that be amazing? It would be amazing. So basically, that is the process of DNA replication. So if I'm half, they cannot come with the left side on my left side to attach me. It's not going to be complementary. So now you know DNA replication is the process whereby DNA makes the identical copy of itself. That is DNA replication. This process occurs in the nucleus during the phase interphase. Interphase is part of cell division process, which we are going to discuss on the other lesson. So now you got what it is, and then where and when does it take place. Why does it take place? Why is it important? Because we want to double the genetic material. Remember, DNA carries genetic material or genetic information. For us to get the double genetic information, DNA replication needs to take place. Now I'm going to summarize the process of DNA replication, and I hope you get it. The first thing that needs to happen, the DNA double helix needs to unwind. So remember, it's coiled and wound, so it needs to unwind. When it unwinds, we are going to end up with a double-stranded molecule, which is not coiled. I hope it makes sense. And then the second step is the weak hydrogen bonds which are holding the complementary bases, they need to break. When they break, the two strands, they unzip. Imagine a zip, they unzip. When they unzip, we are going to end up with two separate strands. Nah? When we have two separate strands, these two strands, they need to act as a template, or they need to act as a guide for the formation of new strands. If we have TGC, we are going to end up with adenine that pairs with T. The new strand should also have C that pairs with G, and then we're going to have G that pairs up with C. So that is the formation of the new strands. And then the same thing is going to happen on the other strand. And then from there, it means you are going to end up with two identical copies of DNA molecules. So they are then going to coil again, or they are going to become helical and coil, and then they will make a double helix again. During this process, the enzyme that is responsible for this process, it's called DNA polymerase. That is DNA replication. That is your 10 marks. So your next lesson is going to be about transcription and also translation, which plays the important role in protein synthesis. I hope you enjoyed this lesson. If you did, please subscribe, like, share, and comment. Do not forget to leave me a comment on the topics you want me to discuss, but make sure that you have subscribed first. Bye.